Welcome back, folks, to part 3 of our section of notes on AP Biology's cell cycle and cell cycle regulation. Here we're going to focus on 4.7, cell cycle regulation. How do we get off this crazy merry-go-round and make sure we just don't keep spinning when we shouldn't? So let's do a brief overview. The frequency of cell division varies with the type of cell. Your epidermal cells, skin cells up here, you're replacing this entire layer every 30 to 40 days or so. So we have pretty high frequency of cell division in these cells. But in other cells, like nerve cells, you have very little cell reproduction. So it depends on which type of cell, how rapidly this happens. And those differences result from regulation at the molecular level. Has a lot to do with cell signaling and also our regulation of the cell division itself. Cancer cells, the reason they become cancerous is they manage to escape the usual controls of the cell cycle and start going crazy. The cell cycle appears to be driven by specific chemical signals present in the cytoplasm. Okay? We don't still know everything about it, but chemical signals in the cytoplasm seem to be the key. The sequential events of the cell cycle are directed by a distinct cell cycle control system, similar to a clock. Kind of regulates how long it's in each of the phases. And it's regulated both by internal and external controls. Okay? And so we'll get into those in a minute. The clock has specific checkpoints where the cell cycle stops until you get the go-ahead signal. So cell cycle goes around and cell's growing, it's doing its thing, and it hits this point. If it doesn't have a go-ahead signal, it's like there's a red stoplight there and it says you can't go yet. And then all of a sudden, conditions are right and it says, okay, keep going, boom, it lets it through and it keeps moving down its pathway. So here we can kind of see the cell cycle. We got interphase in the green and the blue, mitosis in the yellow. We can see here is a barrier in the G1. Here's another barrier between G2 and M, and another barrier right in the middle of M. Those are the major checkpoints. So let's get into it a little bit deeper. Okay. The cell cycle clock seems to be regulated a lot by two main regulatory proteins, cyclins and cyclin-dependent kinases, or CDKs. Okay. The activity of a CDK rises and falls with changes in the concentration of its cyclin partner. So cyclin concentrations go up and down. The cyclin-dependent kinases, they're always there, but they can only get activated when you get enough cyclin partner to bind with them. MPF is one such complex that triggers a cell's passage past the G2 onto the M phase. So it's this one right here that we're talking about right now. And they have similar things for these ones. So let's take a look here. So here's that cell cycle. Here's the cyclin-dependent kinase. And the cyclin its partner is in purple. So cyclin-dependent kinase we see is always here. Cyclin concentration will start to rise and rise and rise, and it gets high enough, it will bind and will form that MPF complex with the cyclin and the kinase together. That will allow things to progress into mitosis, but it doesn't stay stable very long. It starts to degrade. It degrades, pops off, and we're back to the kinase here, and we got to rebuild our cyclin amounts. Over here, we can see the same type of thing. Our cyclin amounts go up, and they get to a high enough point. They bind, and we get the MPF activity. We get the bound thing right here. But then we start degrading it. Boom, once it's all degraded, no activity, nothing happens. Then we start to build up our cyclin again, and it happens. So this is kind of like a delay fuse. It takes a while to build this back up. Once we get high enough, boom, it does its thing. It takes a while to build it back up. Once it gets high enough, boom, it does its thing. So these stop and go signals come from both internal and external signals at the checkpoints. Many signals registered at checkpoints come from cellular surveillance mechanisms within the cell. Basically, the cell is saying, is everything ready to move on to the next phase yet? Do we have all the parts we need? Is everything organized right? There's no sense in getting started if we can't finish, kind of stuff. But you also get outside checkpoints. Is this tissue ready to grow and repair itself? Or does it need to grow and repair itself? You don't want it to grow when you don't need it to grow, right? You don't want your ear to grow when you don't need it to grow and get Dumbo sized. There are three big important checkpoints in each of the cycles, G1, G2, and M. For many cells, the G1 checkpoint, the one that happens at first growth phase and interphase, seems to be the most important. If the cell receives that go-ahead signal for G1 checkpoint, it will usually finish the rest of the cycle. It's rare that it will get stuck for too long and not finish its cycle. Okay, and it will get to M and divide. If the cell does not receive the go-ahead signal at the G1 checkpoint, it's not going to continue on that path of division. It's going to exit the cycle and kind of switch to a non-dividing state we call the G0 phase. Basically, it's in pause mode. It says, just live your life, be a cell, do your thing, make the hormones or whatever it is you do for the body. We don't anticipate needing you to divide anytime soon, so it's in pause mode. It doesn't continue on to where it starts into synthesis, where it replicates the DNA and then moves on into mitosis. So here we can see that. Here's that G1 checkpoint. It gets here. If the cell's not going to need to divide for any reason, it just gets off that merry-go-round, goes somewhere else for a while. But eventually, or if we need that cell to divide to repair some tissue or grow or do something else, we can get a go-ahead signal that drops that barrier, and the cell can continue on into synthesis and start replicating DNA and getting ready for division. Another example of an internal signal is that cells will not begin anaphase until all the chromosomes are properly attached to the spindle at the metaphase plate. So remember, metaphase, you got all of the chromosomes lined up in the center, and you got the mitotic spindle attached to both sides, so you can pull the sister chromatids apart, tops and ends of the cell. If that's not all ready to go, and you trigger going into anaphase too soon before you've attached the mitotic spindle to both sides of that kinetic core, those sister chromatids, you might wind up pulling both sister chromatids to one side. So this cell has an extra chromosome, and this cell over here doesn't have it at all. Okay, and that would be disastrous for both cells. So we have that checkpoint there, and ensures the daughter cells have the correct number of chromosomes. So it won't progress to anaphase from metaphase until everything's ready to go. Everything's lined up, everything's attached, everything's good. If it is, it drops the barrier, and we can continue on and finish our division. That's why you kind of see it here in the middle. So those are internal factors that are there just to make sure everything is ready to go. You've got enough of stuff, it's in the right position, those kind of things. We have external factors that influence cell division. Some of those we call growth factors, which are released by certain cells, which stimulate other cells to divide. So we've got density-dependent, density-independent inhibition. So density-dependent inhibition, crowded cells will stop dividing. So if the cells are all touching each other and they're crowded in there, that means you got enough cells, no sense dividing, so it stops the division. On the other hand, if you're not dense enough, like say you got a cut or a tear or a scrape, and all of a sudden we don't have cells there, it's saying, hey, there's nobody next to me. I can grow that way. And then, so that's density dependent, and it will grow to fill that gap. Okay? So once it's full, stop dividing. Everything goes into G0. If it's not full, let's fill it up. Let's repair that wound or whatever it is. Okay? There's also anchorage dependence. To divide, cells must be attached to a substratum, to something to grow on. This assures that things grow where they should. So you got to have a surface there. So density-dependent inhibition and anchorage dependence check the growth of cells at optimal densities and locations. Cancer cells exhibit neither type of regulation. They'll grow wherever, and doesn't matter how dense they are, they want to keep on growing. So put it all together here. Anchorage dependence, they need a surface to grow on, and they'll keep growing until they fill up the space. If there's a gap, they'll grow to fill the gap, and that's normal. And that's how we grow, and that's how we repair tissues. Cancer cells don't do that. They just grow all over top of each other, anywhere they want, and keep on going. So let's take a look at that. Loss of cell cycle controls in cancer cells. Cancer cells do not respond normally to the body's control mechanisms. And there's a bunch of different ways that they don't respond normally, depending on the type of cancer. They may not need growth factors to grow and divide, right? They may make their own growth factor instead of relying on it coming from other cells. They may convey the growth factor signal without the presence of the growth factor. So, you know, there's an on signal stuck on somewhere in the transduction pathway, or in the cell receptor, the ligand receptor for the growth factor. Something may be there and it's just stuck in the on position. Um, or there may be some other abnormality in the cell cycle control system. A normal cell is converted to a cancer cell by a process called transformation. That's when it goes from being normal and dividing like it should to not. Cancer cells that are not eliminated by the immune system, and trust me, our immune system does a really good job of getting rid of cells that are dividing abnormally, but it doesn't always catch everything. Um, okay, so if they don't get eliminated, they form tumors, which are masses of abnormal cells within otherwise normal tissue. And if those abnormal cells remain only at the original site and don't spread out to other places in the body, we call them a benign tumor. Okay, come back. Malignant tumors, on the other hand, invade surrounding tissues and can metastasize or spread to other parts of the body where they can form additional tumors. If your tumor is localized, we can treat it with high energy radiation because we know exactly where it is. We can pinpoint it. We can kill it with that radiation and destroy the damaged DNA in the cancer cells. It's probably going to affect the cells around the tumor as well. But since we know it's a local spot, those can grow back in, the healthy tissue hopefully grow back in to replace it. Now, if the cancer has become metastatic and it started to spread elsewhere in the body, then that localized treatment, either cutting it out and or hitting it with the radiation, isn't as effective. We have to add in chemotherapies, chemicals, which target the cell cycle to try to stop this. They target the cell cycle. We try to make them as specific to the cancer cells as possible, but they generally still have some effect on the rest of your cells, which is why people on chemotherapy have a really rough time. Like they're exhibiting the signs of these cells not dividing properly. So here, just a picture to illustrate that. There's a tumor in the breast glandular tissue. Um, Cancer cells invade neighboring tissues. If they get big enough, they get close to a blood vessel or a lymph vessel. Now cells can break off and go elsewhere in the body, and sometimes they can lodge somewhere and start growing more, and you get new cancer tumors, and it's metastasized. And that is it for our notes on this unit, guys. If you have any questions, make sure you ask. Talk to you later.